In this episode of Voice of the Sea, we're in the murky waters of Kāneʻohe Bay with researchers from the Hawaiʻi Institute of Marine Biology who are studying the response of fishes to muddy water. Runoff from land into the ocean is a natural phenomenon, but human-occupied areas can have much more, up to 10 times more sediment than would naturally occur, choking our nearshore fish habitats. ʻāina stewards are working to bring back abundance to nearshore reefs by restoring streams and wetlands, lowering the amount of sediment released into the ocean. But how clean does the water need to be? What level of sediment causes herbivorous fishes to flee, leaving corals vulnerable to overgrowth by algae? The Johansen Fish Resilience Lab is monitoring how water quality and reef health has improved in certain areas, thanks to the revitalization of nearby wetlands and loko ʻia. The lab is also working to establish tolerance limits of reef fishes to sediment runoff and develop water quality thresholds to keep our nearshore fish abundant. First, we head out on the water with Professor Jacob Johansen and researcher Linda Egertsen.
So we're heading to two of our sites that we have been surveying for a while. One is quite sedimented, muddy, still to this day. The second one was one of the worst sites and has since started cleaning up with work from the community. And we are seeing real improvements in the fish abundance. So we'll see the two contrasting sites today and what cleaning up the water can do. We're at one of our most sediment inundated sites. So they're doing surveys as who's here, what does the benthos look like, what is the water quality, how much sediment is coming into the water, what's the turbidity, so that's the visual clarity, and what's the sediment settlement rates, how much is it covering the benthos, and then what algae are there, and then the fish that are here, what are they doing?
Before we jumped in the water at the murky site, we took a look at the tools that the team is using to measure water quality, fish abundance, and overall reef health at each location. So you have some tools and stuff with you today?
We do. We also have this probe here that measures salinity, water temperature, turbidity, so we can have that continuously along the day. We measure light conditions, and this here is a sediment trap. It doesn't look very advanced, but...
How does it work?
We take these caps off and then leave it on the reef for a couple of days. So the idea is that sediment settles and just snows down kind of, and then you get a volume and surface area, so you can calculate how much sediment we get on a certain area on the reef. So we compare the different reef sites with that. We do have a light meter, so this measures light on the reef, so we can, because that's also one of the main things that gets affected by turbidity.
So when we get in the water right now, what are we going to do?
We're gonna mount these things on our experiment setup. We also have fish surveys going on. That's the tape, another scientific equipment behind you, to count fish abundances at the different sites.
We're going. After seeing the murky study site, we traveled south in Kāneʻohe Bay to snorkel the reef near ʻāina loko ʻia. Recent removal of invasive plants and stream restoration is helping to filter sediments and clean the water, resulting in noticeably healthier fish and coral communities on the nearshore reef. So that was a pretty dramatic comparison.
Yeah, it's very different. When we started working on this project, the lab chose this site just because it was one of the worst sites. It was really murky. And along the time of the project, the improvements made in the fish pond has actually helped stop a lot of sediment coming out, which is really positive because we see these really nice changes in water quality, and what we saw today on the reef too with all these fish and a lot of live coral. That's a really nice improvement. We do see that the worst site we went to this morning had really low fish abundance and biomass. If you were to fish there, you wouldn't really get that much to eat that day. We also see that intermediate levels of sedimentation already have a really large effect on abundance of fish and diversity. And there's lots of fish that will not be present on those sites. And then when we come back to this site, which is really nice, we can see that we get back most of those. Some species seems to be more tolerant towards sedimentation, and some are really sensitive. We wouldn't see unicorn fish, for example, and a couple of other species that would be really hard to see them on even when there's a little bit of sedimentation. So another effect that we're seeing from the sedimentation is also that we do see a lot of invasive species on these sites. That was somehow unexpected, but it seems like turbidity facilitates for some invasive species to actually be able to establish themselves on these reefs. We do have the toa, which is an invasive snapper. We see a lot of them in turbid waters. And there's also, as we saw on the first site, a lot of invasive algae.
Historically, or sort of anecdotally, the theory was the sediments come in, they settle on the coral, they basically kill all the benthos, and so the fish leave. The management strategy for a long time has been really to preserve the fishes that can eat the algae that will then allow the corals, or give the corals a fighting chance. What we are now seeing is that that's probably reversed. It's actually that the fish are leaving, and then there's nothing left to protect the corals, and so the whole system goes down. The sediment that comes in can affect fish in many ways. It can clog their eyes so they can't see any of the food or the predators they're trying to avoid. It can clog their nostrils and epithelia so they cannot smell. A lot of these fish find their food through smell, so that's gone. It can clog their gills so they cannot breathe. And then basically it can cover their food. So they can't see, they can't smell, they can't breathe, and there's no food. Which of these drivers is actually the issue, and is there a separation between some species being more tolerant than others? Then you can focus your management strategies to those as sort of like an indicator species. So we have surveys going on here, looking at what species are here, what is the composition of the different herbivorous fishes. So we have our scrapers, the ones that basically scrape off the substratum, removing even anything that's there to then leave more substratum for corals to settle. Then you have your grazers. Those chop down the little turf algae that would otherwise start to overgrow and block out or kill the coral. And then finally you have your browsers. They eat the big macroalgae. Once it's established and the system has changed, you have big macroalgae. And in order to get those down and even give the system a chance to recover, you need those chopped down. And if you want a functioning system, meaning you need groups from each of these three herbivorous functions, is there a difference in sensitivity for each of these three groups? Then what are the water quality levels you need to get to in order to see improvements? That data doesn't exist, and that's what we're coming in with. The world over, there's an agreement that water quality is a problem, it's killing coral reefs. But there is no hard, fast numbers to give a manager, to give a community, to give anybody to say, you need to get your water quality to this level in order to improve things. Well, how much work is enough? What level of water quality are you working towards? That doesn't exist. So if you're telling a community you need to clean things up, they're like, great. By how much? By what level? We've been working on and some of these communities have been working on their water quality for 20 years, and it is better. But you may not see any improvements because you haven't gotten to the threshold beyond which you start having recovery of fish. If you're a community, if you're a management team, if you're trying to improve things, what are the levels you should be working towards? We can see that if you do clean up the water quality, very rapidly, as soon as the fish can, they are moving back in. So when you start clearing up, you'll get 400-500% more fish on these reefs. So we're seeing major recovery in sites that are now being improved by the community. So I'm very excited that we're getting to a point now where I believe we actually can help improve the inshore reefs majorly. and provide more food security. In a time of rapid degradation of coral reefs globally due to bigger issues, this is actually something that can help local communities, and I'm excited for that.
The University of Hawaii's Sea Grant College program focused on Hawaii's coasts and its communities through sustainable development, safe seafood supply, sustainable coastal tourism, hazard resilience, and healthy coastal ecosystems. Hawaii's Sea Grant.
You're watching Voice of the Sea. We're talking with mathematical modeler Lisa McManus about how math can help predict the thresholds or water quality levels needed for fish abundance and reef health. But first, we head to the lab with graduate students Jasmine Reichard and Matthias Schachman to learn about the fish behavior experiments that provide data and insight for the mathematical models.
This is one of the experiments that we have set up to kind of look at the behavioral responses of some of these herbivores to sedimentation. So as you can see, we got these small little tanks. There's a little false bottom, and then we got these little holes that run through. Between each of these tanks, there's two lasers, infrared lasers, that gets triggered whenever the fish swim through it, and that's then connected to a computer. So this is the clean tank. And then as you go through, you can see it gets more and more sedimented. And at the end, you can barely see down to the bottom.
What kind of fish is in here now?
This one is the ringtail surgeonfish, Acanthurus blockii. It's one of the fish that we see them more often on intermediate to high compared to other grazing fish. So I want to see if his threshold actually is higher than the other fish. And that's kind of what I'm expecting to see.
The computer calculates for you how much time the fish spends in each one of the tanks.
Exactly. So we got this homemade program, and you can see the bars are just the tanks, and then it just counts how much time it spends in each tank. It's relatively simple, but it's very powerful to figure out where the fish wants to be.
And then this graph is showing you which tank it spent the most time in. Oh, he's coming back.
Yeah, now he's swimming, should be in this tank. From all these surveys we've done out in the field, we see that we very quickly lose a high amount of these fish, even on low sedimentation levels. We don't have to get very far, and we lose 80 to 90% of the most important fish we need on these reefs. We want to see, is this because of the sediment? And particularly, can we detect a level in which they completely avoid? So we can kind of relate and validate these field observations we see in the lab. We're actually also running some of the invasive species that we do see out on the impacted reefs. And we see that they actually somewhat prefer the sedimentation. Specifically for the ta'ao, the black-tailed snapper, which is one of the most abundant invasive species we see on some of these reefs here in Hawaii. And they love it. They just venture into all the sediment. They don't really care about it, essentially.
Are you guys actually engineering and building all the experiments yourself?
Yes. It's actually one of my favorite parts of doing what I'm doing is, with marine science, there's so many other things you're gonna be able to do. Engineer, like design, carpentry. This particular experiment I put together over a year. It took a long time to set all these tanks together and figure out how the laser system and programming. You just got to figure it out.
Yeah.
Get in and try to build it.
So here we have our foraging experiments that we do on all of our herbivores. We do this in the field and then in the lab. It's kind of how we cross-validate what we're seeing out there, and also a way for us to get exact levels of sediment by manipulating it in the tanks. Here we have our unicorn fish. This is our control. There's no sediment added here. We have our low treatment, our intermediate, and our hide down there.
So these on both sides are doing similar studies, but with different fish and types of feeding.
Yes, exactly. So this is our scrapers, our parrotfish, and then these are our browsers that eat the macroalgae. We're working on multiple species.
We have to do one at a time.
Yes, one fish at a time goes in each of the tanks. So it's a lot of experiments.
Right.
Yeah.
That's why a lot of time in the lab.
Yes, a lot of time in the lab.
This is the first step into developing what we hope will be eventually a decision-making tool. You know, one of the tools in the toolbox. What might come out of this is even something like, depending on what types of herbivores are in an area, maybe there's a different sedimentation threshold, right, that could be set.
Show me behind the curtain, like the graphs are super cool.
Yeah.
But what does it look like when you're working on it?
Yeah, well, here is one example of what it looks like. Basically, this is the set of equations that we use, and then we build the mathematical equations, and then we write a lot of code to be able to then run these simulations and eventually get at. Here is an example system where as you increase the sediment concentration, everything's dropping. So these scrapers in blue, the browsers in red, and the grazers in yellow are all declining. And here you are seeing this spike in turf algae, and the coral is dropping. And then all the way, when you have high sedimentation, you have very high macroalgae. And in this model, basically what's driving that is that there is no longer anything eating the macroalgae if you get rid of the fish. Jacob and I are collaborating with the education faculty here at HIMB to develop modeling modules. This will be another module that hopefully gets students excited about combining math and computer science to study ecology. And it'll be cool because then they can see how it all fits together. The math, the code, you know, these figures, they all connect to real things that are really important to us.
We are looking for a few heroes, mentors, trailblazers, innovators, a passion to change lives, spark curiosity, open hearts, and awaken minds, help students answer the question, who am I? This could be your calling, but this is no job. It's the journey of a lifetime. Be a hero. Be a teacher.
You're watching Voice of the Sea. We're at Kāneʻohe in the wetlands above the Heiʻia Loko Ia, talking to scientists and ʻāina stewards Shimi Ree and Yokepa Minor, and Division of Aquatic Resources Fellow Ellie Jones, about how their ʻāina work and monitoring programs are improving not only the health of the wetland and the lo'i, but also the health of Kāneʻohe Bay.
I grew up here in Hawaii, and I wanted to do science and research that impacted the people here. My role is to coordinate and facilitate research in the Heiʻia Ahupua'a. The sensors that we put out, that's taking measurements all the time. They measure temperature, salinity, dissolved oxygen, pH, and turbidity. And then some of them have chlorophyll sensors to look at productivity. Those are taking measurements every 15 minutes. But we do come out every month and then also take grab samples of water for nutrients. And then quarterly, we take DNA samples. We look at microorganisms as well as what sort of fish that we might not be able to see that's, you know, that are lurking in the water. Usually, if there is a big dumping rain, then we would actually get high turbidity for just a little bit, and then followed by that is chlorophyll, a little algae bloom or something. But it goes away very quickly.
When you talk about high turbidity and then an algae bloom, are you talking hours or days?
That's kind of what's been interesting about having a sensor in the stream mouth. It doesn't last very long. It's a very dynamic system, so it just moves along in about maybe a day or half a day.
Have you been able to detect any change over time as Kāneʻohe has been restoring the land and helping the water to find its path.
Now, this whole place that we're sitting in used to be all mangroves. One of the questions that we kept getting was like, if you cut down these mangroves, are there going to be tons of sediment going into the nearshore reefs? In collaboration with a lot of HNB scientists and DAR and TNC, the Nature Conservancy, we did a study and we found that actually turbidity and sediment trap data didn't increase. And it's really because of these waterways and the lo'i. They serve as sediment retention basins to prevent sediment from getting into the nearshore reef. And at the same time, they provide habitat for native species. It's really important to understand what is natural variability so that if something different happens, then you know that's outside of our natural range. Do we need to do something about it?
Talk to me about the water that moves through this Kāneʻohe, ʻāina.
You know, there's pieces of the water in Kāneʻohe we're still trying to figure out. For us, in the time that I've been here, it's just been trying to tap back into those waters, slow down the straight shooting, and let it kind of meander and curve where it naturally wants to. I seen the first native plants that popped out after the mangrove drop. And then because we opened up some of the first waterways to go back under the bridge, kind of knew where to look, to watch as things might start to come upstream. And then as for the birds, now we have thriving families of these birds and they're naturally coming back. More coming, birds that didn't exist here 5-10 years ago. We have some places that it's so dense with native species that we don't have to manage it 100%. The ahupua'a function is present. It's functioning as it should. Now that we have this open water to see the water, not to see a biofilm sitting on top, to just see vai. To have the vai be here as the current reminder, like how vai this place is.
This is a really wonderful place because it's given us such an example of how wonderful restoration can be done when it's done well and when it's led by community. And they've got these projects going all the way from the ridge down to the reef. And so you get to see how these estuaries can act kind of as sponges for water quality before it gets out onto the reef. And so it's kind of collecting a lot of that sediment, collecting a lot of that water that might have excess nutrients before it gets out onto the reef. The question I've been trying to answer is, what is DAR's place in the water quality world here in Hawaii? Since this is not something that we're mandated to do, but it's something that we've been hearing from the community and something we've been hearing from fishers, and so it's become a priority for us to try to figure out how to incorporate that.
Why?
Because it's something that we're seeing is degrading reefs. We have a little bit less information here about how it's affecting fishes, which is why some of the research that Dr. Johansen and his lab is doing is so important, because we need that type of information to be able to make decisions.
How do people get involved?
If you're interested in getting involved or learning more, you can go to our website. Every once in a while, we'll also hold public meetings to provide input, and so those will be posted on the website. And if you ever see us out and about in the water or somewhere here, just come up and ask a question, say hi. I'd love to talk.
Runoff and sediments from land are problematic worldwide, reducing fish populations and killing coral reefs. But local action makes a big difference. You can get involved in restoring local fish abundance and reef health by volunteering at coastal cleanups in your area. Learn more about water quality, fish behavior, ʻāina restoration, and community projects on our website at voiceofthesea.org. Follow us on social media at Voice of the Sea TV. Mahalo for watching Voice of the Sea. Exploring Our Fluid Earth is the dynamic curriculum developed by the University of Hawaii's Curriculum Research and Development Group. The award-winning Fluid Earth and Living Ocean textbooks are now interactive and online. New activities, updated content, and a teacher community. Exploring Our Fluid Earth is now freely available. Find out more at exploringourfluidearth.org.